Would you want to permanently fuse to your significant other? Probably not. But reproduction can have evolution acting in some pretty weird ways. Let me tell you about 2 ways nature has evolved to optimize reproduction by allowing living things to coexist while physically connected, and how this can give us insights into preventing organ transplant rejections. We will also be hearing from an expert, Dr. Jessica Weaver, a biomedical engineer at Arizona State University, working on novel methods of immune tolerance. Thank you, Dr. Weaver. Let's begin with the strange reproduction of deep-sea anglerfish. There are species of deep-sea fish where the male fish can permanently attach to the females after reaching puberty, like actually become part of the female's body, and can even integrate their blood vessels. It's an odd lifestyle choice, but hey, I'm not judging. It's an adaptation for living in the deep, deep part of the ocean, where it's dark and devoid of life and hard to find a mate. I guarantee you, if you found someone down there, you wouldn't want to let go. Once attached to the males reduce in body size substantially and lose their ability to feed or survive on their own, instead receiving nutrition via blood circulating from the female. Now let me tell you, this is unusual. If I attached to you and our circulatory systems fused, becoming one, we would both die immediately. This is because we have adaptive immune systems, which means that our immune system identifies what is self versus non-self by recognizing proteins on the surfaces of our cells called antigens. One class of antigens important for successful organ transplants is MHCs. MHC molecules are highly specific to an individual. So my MHCs are different than your MHCs. And this is the reason for why, with respect to organ transplants, you can't just get an organ from any rando. Instead, you need to find a match, a person that has similar MHC molecules to you, and even then, you will still probably need to take immunosuppressant medication to dampen the immune response so that your body does not reject the organ and kill you. One exception to this is that if you get an organ donation from an identical twin, then you wouldn't need immunosuppressants. Your MHCs, like the rest of your genes, would be identical. Well, okay then. So how do the fish do it? How are they okay with fusing 2 separate beings into one and, not dying from the whole incompatibility of their MHC proteins thing? Well, there are 2 possibilities. Number 1, that their MHC proteins are all similar enough that they can fuse to each other and not elicit an immune response. Think of the twins. Or 2, that these fish are lacking proper immune responses. This has been puzzling scientists for a long time. However, a recent paper showed that these fish are lacking major components of their adaptive immune systems. This is very surprising because all jawed vertebrates have an adaptive immune system. It's a defining characteristic. The adaptive immune system, like the innate immune system, serves to protect my body and your body from foreign compounds and pathogens, with the added component that it, adapts. These immune cells learn from previous encounters with a given pathogen and are prepared for the next encounter. What types of immune responses are lost in these species? Well, it does depend both on the types of attachments and the number of attachments each species undergo. Some species only form temporary attachments, while others form permanent attachments, i.e. the whole fusing blood vessels and staying for life thing. And some of the species that form permanent attachments have 2 more subcategories. Either they form one-to-one relationships, or it can be many-to-one, where there are multiple males attached to each female. What is interesting about this is that there are fewer changes to the immune system in the species of anglerfish that have temporary attachments, while there are more changes to those who do the permanent types of attachment, and even more changes to the ones that do the many-to-one permanent type of attachments. I mean, just consider this. In the species where many males permanently attach to one female, there is a loss of genes that leads to the disruption of both T and B cell immune responses, whereas in temporarily attaching species, there are only genes missing involved in B cell pathways. But why do these fish not need adaptive immune responses? Are there no pathogens in the deep sea? That's not quite it. The researchers think it could be that these species have instead reorganized or revamped their innate immune responses. See, not all animals have adaptive immune systems. As I mentioned before, only jawed vertebrates have adaptive and innate immune systems, while all other animals only have innate immune systems. So it's not necessary to have an adaptive immune system to survive. However, the innate and adaptive immune system in vertebrates has been evolving together for the last 500 million years and was thought to be an intertwined system that relied on both components. Therefore, The loss of one component would be detrimental. However, these fish challenge that view, showing that the loss of the adaptive immune system is indeed possible. This has implications for understanding how the innate immune system could be enhanced to help patients that have compromised adaptive immune systems. So, there are no other examples in nature where two separate individuals fuse like this? Well, that we know of, and that's a pretty big caveat. But as far as we know, these species of fish are unique in this sense. However, there are other circumstances where a foreign organism attaches to another and shares a common nutrient supply, including one very common phenomenon called pregnancy. A fetus is made up of combinations of its parent's DNA, so it expresses maternal antigens which are foreign to the mother's body. In instances where mothers later in life receive organ transplants from their children, they still require immunosuppressants to prevent rejection. Yet, the body normally does not reject the fetus during development, all the while without making use of immunosuppressants. How is this possible? It's because of the placenta. The placenta is a really fascinating organ. It is a recently evolved organ and a temporary one. The placenta has many roles in pregnancy, including supplying the fetus with nutrients and gas exchange, as well as training the mother's immune system to tolerate the fetal tissue as self. Researchers such as Dr. Weaver are working towards using the immune tolerance techniques of placental cells for organ transplants in humans instead of immunosuppressant drugs, which have a lot of non-specific effects and cause a variety of bad side effects. So how does the placenta promote immune tolerance, and what does this mean for the future of transplants? Recently, April interviewed Dr. Weaver, a biomedical engineering professor at Arizona State University, working on using the immune tolerance abilities of the placenta for applications in organ transplants.
Hi, Dr. Weaver. Thank you so much for joining me today. So I see you won an Innovator Award for your insights to use the placenta as a model for immune tolerance. Can you tell us more about how you got involved in this type of research?
Yeah, great. Well, thank you for having me. My background is actually in islet transplantation, which is a cell therapy to try and treat type 1 diabetes. The issue with trying to use a cell therapy for type 1 diabetes is that the risks of immunosuppression vastly outweigh the benefits for most of the patients with type 1 diabetes. So that really led me to focus a lot on the problem of the immune system and the problem of immunosuppression. And we did some really cool work trying to deliver molecules that could potentially help hide grafts from the immune system. And one thing that I noticed is that a lot of people focus on these mechanisms that we've discovered in cancers. We know what signals cancer uses to hide from the immune system. One thing that stood out to me about that model is that cancer is your own tissue, right? So cancer can hide from the immune system, but it's a really low bar to hide from the immune system because you don't have that tissue mismatch, so the MHC or the HLA mismatch. I tried to think about any sort of biological examples that we have where we truly have a foreign tissue that's not attacked by the body. That's sort of how I landed on pregnancy, which is the only biologically natural example of where our body would accept foreign tissue as self. And so I started digging into that. I didn't see anybody really working in that space or working with the placenta. And as I looked at pregnancy more and more, it's funny, only in the last, I want to say, decade or so has the research really delved into the mechanisms of how pregnancy proceeds without a hitch. And we used to think that pregnancy was a state of immune suppression, but now we know a lot more, and we know that it's actually the placenta, which is derived from the fetus, induces an antigen-specific tolerance in the mother. So basically trains the mother to believe that that tissue is her own tissue.
That is all really cool. So how does the placenta do it? Like, what are the mechanisms that it uses to induce this immune tolerance?
The placenta uses many multi-layered mechanisms to induce this antigen-specific tolerance. So one of the primary ways that the placenta induces tolerance is the cells of the placenta, which are called trophoblasts. These are specialized cells, and I like to call them professional tolerogenic cells. So one way that the trophoblasts do this is they actually don't have most of these classical HLA. And what's interesting is they have these other HLA that are called non-classical HLA, like HLA-G and E, which are still not really super well understood. But from what we do know, it appears that HLA-G and HLA-E contribute to this tolerance. So that's one mechanism, so through that direct contact. But we also know that trophoblasts are spitting out a ton of signals that are telling the maternal immune cells, do not recognize this as foreign. And so it's this combination of the proteins that are presented at the surface of the cells interacting with the maternal immune cells, and then the signals that they secrete, and even how they actually decorate their proteins. And what's interesting about pregnancy is these multi-layered mechanisms are so robust that you can delete one important pathway or another and not cause pregnancy loss.
It's really amazing how robust these biological systems really are. So what does this work mean for the future of organ transplants?
I mentioned that my background is in islet transplantation, so basically transplanting cells. Transplanting individual cells makes it a little bit easier to engineer solutions than it is to try and address whole organ transplantation. So when we're delivering cells to a transplant site, we're not taking the islet cells and putting them back in the pancreas. We usually put them in a different site, like under the skin. A lot of people like that location. But basically, we make like an ectopic organ. And because of that, we can do a lot of localized solutions. Take some of these immunomodulatory molecules and present them locally in the transplant site. And that was pretty successful at reducing immune responses against those cells. And again, so you don't have this systemic immune suppression, you just have this trying to induce a localized tolerance against the cells. The hope is to then translate things that we develop into strategies for organ transplantation. One thing that we've started with is taking these extracellular vesicles that the trophoblasts secrete, because they're like huge packages of information, lots of tolerogenic molecules. Taking those extracellular vesicles and developing a localized drug delivery system within the islet transplant site to deliver those extracellular vesicles. We're also developing a method to deliver trophoblasts basically as a cell therapy. We have our insulin secreting cells to try and treat the type 1 diabetes, and we envision delivering tolerogenic inducing cells alongside them to work as kind of a long-term local delivery mechanism to induce tolerance against the graft. But we're not even sure that we would need to have, these signals delivered for the lifetime of the graft. Because what's really interesting about pregnancy is, at least in mice, we've seen that the more pregnancies that you have with the same father, so the same paternal antigens. Let's say you have 2, 3, 4 pregnancies. With each pregnancy, the tolerance becomes more robust and kind of more permanent. And so we see this hyporeactivity against paternal antigens after multiple pregnancies. So that points to the idea that we could potentially train the maternal immune system for long-term tolerance against these antigens without having to have like a continuous signal alongside the graft. And so that would really help us translate this to whole organ transplantation, because what we could do is create essentially a vaccine, a tolerogenic vaccine, where if we deliver the antigen for the whole organ, take some of the donor's cells or antigens, deliver it with some of these tolerogenic molecules to produce this robust tolerogenic response against those antigens, then you could potentially transplant the organ without having an immune response against the organ.
Okay, so this all sounds really cool. So I have to ask, how far away is this type of technology from being used in the clinic?
I don't like to overpromise. This is probably a couple of decades out. I'm not going to lie. It's a very new idea. It has to go through a lot of, first of all, we have to test it and see if it even works. We're in the very early stages of investigating how we could pull this off with trophoblasts and their mechanisms. And so it's very early stages. We have to go through all the preclinical testing, safety, and then, clinical trials. But, I think obviously it would produce just huge advantages over current techniques.
Well, thank you so much for coming and telling us about your research. I really look forward to hearing more about your work in the future. So thank you again, Dr. Weaver.
As with many things in nature, something as wild as 2 individual organisms coexisting while physically connected manages to work out and even become an integral part of reproduction for many species. In medicine, we can imitate these mechanisms nature uses for our own advantage in things like organ transplants. And whether 2 organisms coexist by pretending to immunologically not see each other, like the anglerfish, or by evolving protective barrier between them, Like in pregnancy,
Life finds a way.